next up in this today's video we will discuss about the transfer RNA. This transfer RNA, it is also known as tRNA or it is also known as soluble RNA. There are different type of RNAs present like messenger RNA also known as mRNA, ribosomal RNA also known as rRNA, as well as this transfer RNA which is known as tRNA. This transfer RNA, it is made up of 75 to 90 ribonucleotides. It is made up of ribonucleotides. This is having different type of structure. This transfer RNA is having different type of structures, like it is having primary structure, secondary structure, as well as tertiary structure. What is the main function of this transfer RNA? This word itself tells that it is helping in transfer. Transfer of what? It is a transfer of amino acid for the formation of proteins. So there is a mRNA and there is a triplet code which is present in the mRNA which decides the formation of proteins, or this triplet code decides which amino acid will come and bind first and which amino acid. In this way the protein is formed. Like we can see here that that is first amino, that is a mRNA present and it is coding like AUG, suppose is a start codon, CAG. In this way that is the mRNA, and this mRNA is coding a specific sequence of amino acids. So this amino acid will come first, then comes the next amino acid, and these two amino acid they will form by the formation of a bond which is known as peptide bond. Okay, in this way multiple amino acid come together to form the protein. So let us see first what are the structures of this tRNA. There is the first structure is a primary structure which is linear like structure. It is having one 5' end and it is also having a 3' end. In this structure it is having certain arms like DR, anticodon loop, T-sic arm, amino acid acceptor arm, and this four reasons is having specific function of each. In case of the secondary structure, this primary structure forms a self-complementary like structure which forms a secondary structure. When the secondary structure is formed, it looks like a clover leaf. That is the reason the secondary structure, it is also known as clover leaf model. It is also known as clover leaf model. The third is the tertiary structure which is known as the 3D structure of the tRNA. It is also like L-shape and it looks like helix stacking. This here we can see that it is looking like helix stacking. Next we will see that in the primary structure we have discussed about certain arms which were present. Here in the secondary structure we can see those arms. Like it is having one amino acid acceptor arm which is recognized by ACC. In this reason, the amino acid will come and bind. And there are 20 amino acid, 20 to 22 amino acid. After the modifications, there are 22 amino acid. And for each amino acid that is specific, there is specific tRNA is present. Okay. Now here we can see that near the 3' end, that is T-sic arm. What happens in case of a tRNA? It is having adenine, cytosine, guanine, and uracil is present normally. But thymine in A complements with U and G complements with C. But there is no T in case of RNA. But due to certain modifications, there is thymine in this tRNA. Due to which here it is shown here that T-sic functions and how they are done. In the 5' end, there is a DR, and here we can see that here it is having a anticodon. Okay, so first we will see what are the unique features of this tRNA. The specific part of this tRNA, it is having a specific sequence which is known as CCA sequence, which is the acceptor arm, and this acceptor arm is not forming any secondaries or it is not complementary with any other part of this tRNA. It is in the free form, and here the amino acid comes and bind, and the bond which forms between this ACC region and the amino acid, it is the ester bond. And the tRNA which are present, they are freely floating in the cytoplasm, and they protects mainly by methylation, like it for the conversion of ionis into adenine, pseudouracil to uracil, dihydroxyurea into uridine. And there are, except thymine, there are other unusual bases like hypoxanthine, thymine, as well as methyl guanine present in case of tRNA. Okay, now this part we have already discussed, that it is forming a ester bond. It is having ester bond, the amino acid, and the ACC part which is bound, it is having one ester bond. And this CCA sequence which is present, it is having one OH group by which there is formation of this bond. Okay, now we will see the D arm. Why it is known as D arm? D stands for dihydrouracil. First of all, we have to understand what is the difference between the uridine or uracil and dihydrouridine. We can see here that there is a ribose sugar present over here which binds with this uridine. Okay, it is bound, this bond between the ribose sugar and this uridine is in the first position and with nitrogen. But after certain modifications, there is two hydrogen groups present in the 6 number carbon and 5 number carbon. These are the extra two hydrogens which are present. That is the reason it is known as dihydrouridine. So what is the, if it is present, what is the function? What is the, if there are certain modifications, if so there is some specific uridine, the function of this DNA, the main function of this recognize a specific site of this enzyme which is known as amino acid tRNA synthetase. So what is this amino acid tRNA synthetase? It is a certain enzyme which help in activating in this tRNA. Okay, in the next video we will discuss how the activation of this tRNA, what is the work of the amino acid tRNA synthetase and how it works. Okay, so this D arm which is present, it is having, we have already seen here that the D arm which is present, it is present near the 5 prime end. We can see here that the D arm which is present, it is near the 5 prime end. Okay, and this D arm is having 4 bases. This is a D arm. It is having 4 bases like 1, 2, 3, 4, which is forming the stem-like structure. It is the stem-like structure, and this is the loop-like structure. Loop-like structure, and this loop-like structure, it is made up of 8 to 12 bases. Okay, this is all about the D arm. It also helps in the stabilization of the tRNA's tertiary structure. Next, we will discuss about the T-size arm. What is the main function of this T-size arm? The main function of this T-size arm is that it recognizes the ribosome recognition site. During the process of translation, there is involvement of ribosome. This tRNA recognizes the site of the ribosome. Okay, so due to certain modifications, the T-size arm which is present, it is present near the 3 prime end. So it is also having a stem and loop-like structure. So in the stem, that is they are having 5 bases like 1, 2, 3, 4, 5. These are 5 bases which are present in the form of a stem. And this is the loop which is made up of 7 unpaired nucleotides. It is made up of 7 unpaired nucleotides. So we can see 1, 2, 3, 4, 5, 6, 7. Okay, these are the 7 nucleotides. Like here we can see that is T. Then here we can see psi, and here we can see C. That is the reason, due to the presence of the T-size, it is known as T-size prism. So first we have to see what is the main difference between the uridine and pseudouridine. In case of uridine, the ribose binds with the nitrogen group of the first number nitrogen group. But in case of pseudouridine, the position of this bond changes. Like now the ribose is not bound with the first nitrogen, it bonds with the 5th number carbon of uridine. This is the only difference of the uridine and the pseudouridine. Okay, so due to this modification, it also protects from certain RNAs which help in the degradation of this tRNA. Next is the anticodon loop. There is an anticodon loop. Here we can see that it is having an anticodon loop. It is also forming stem as well as loop. In the stem, there is 5 bases like 1, 2, 3, 4, 5. This is the 5 bases in the stem in case of anticodon. And in this loop, it is having 7 base pairs. 1, 2, 3, 4, 5, 6, and 7. There are 7 base pairs. This anticodon loop which is present, it is having this three portion. It is complementary with this mRNA. Like we can see that here, if mRNA is present and each triplet of this mRNA is coding for some of the amino acids. AUG is the start product. The tRNA comes and bind in this AUG region. There will be specific tRNA which is having in the anticodon region, they are having a complementary sequence to AUG. That is U A C. Okay, and this tRNA brings the amino acid which is known as methionine, which is known as methionine. So already we have discussed the three parts of this tRNA like anticodon loop, PCIC loop, and D loop. The next is the amino acid acceptor site. Here we can see that it is having a amino acid acceptor site which is known as ACC in the 3' end. Here the amino acid can come and bind, as it is having a OH region over here. It forms a bond between this amino acid. It is known as ester bond. It is known as ester bond. This is all about the function structure of this tRNA. Now the other thing which is present is a variable arm. So, due to the presence and absence of the variable arm, again this tRNA can be differentiated into class 1 tRNA or class 2 tRNA. Here we can see that there is a variable, here there is a outgrowth like structure. This is known as variable tRNA. Here it can vary the number from 3 to 21. Okay, this varies from 3 to 21. So what does the work of this variable arm? It recognizes the amino acid tRNA synthetase, and if there is a variable arm, then it is known as class 2 type of tRNA. If there is no variable arm, then it is known as class 1 type of tRNA. This is all about the structure and function of this tRNA, and if you like this video, please do like, share and subscribe. Thank you.